Begin by sterilizing your hands with 70% ethanol. Next, grab a permanent marker and use the shot plate holder as a guide to make a circle in the center of the medium plate. Light your Bunsen burner and sterilize your forceps. Begin to transfer the rice cali to the osmotic medium, working your way out from the middle. Once the shot plate circle is full, containing about 50 rice cali, wrap the plate with micropore tape and store in the dark until you're ready to shoot. Next, ready your supplies for the transformation. You'll need a sonicator, a biohazard bag, a vortex, the gun parts, a PDS 1000, a vacuum pump, pressurized helium, a microcentrifuge, your rupture discs, stopping screens, macro carriers, 200 proof ethanol, sterilized type 1 H2O, your micro carriers, some sterile plates, your plasmid of choice, some calcium chloride, spermidine, and 2 microcentrifuge tubes. You'll also need a spatula and a scale. Next, begin to prepare your microparticles by sterilizing with ethanol and washing with H2O. While the microparticle preparation is far too detailed to explain in depth, the general process consists of adding the plasmid DNA to the pellet. The plasmid DNA is at a concentration optimized for single copy insertions. After the plasmid DNA is added, calcium chloride is added, followed by spermidine, and then placed on ice for 1 hour to allow adhesion to the microparticles. While waiting for the microparticles, make sure the rest of the equipment is sterilizing in ethanol. Make sure to spray down the trays as well as inside of the gun. After about 45 minutes, pull the equipment out of the ethanol and allow to air dry. Once dry, grab the macro carriers and put them on the macro carrier holders. Use the punch to push the macro carriers firmly into the macro carrier holders. After the 1 hour mark, resuspend the microparticles in the sonicator and turn off the laminar flow hood. Place 10 microliters of the microparticles on each macro carrier. Make sure to resuspend the microparticles in between each aliquot. Allow for the ethanol to evaporate and the gold to dry on the macro carriers before turning the flow hood back on. Next, pressurize the helium line and turn on the vacuum pump. Lastly, turn on the PDS 1000. Now we're ready to shoot. Place the stopping screen in the macro carrier holder and place a macro carrier upside down. Grab the ring and tighten firmly. Now grab the rupture disc retaining cap and place a rupture disc, still wet, inside the cap. Screw the cap on to the PDS 1000, making sure to firmly tighten. Now place the macro carrier launch assembly inside the gun, followed by the target plate shelf. The exact target distance will vary. Close the door and latch. Pressurize the chamber by pushing up on the middle button. Once the chamber has reached 26 inches mercury, hold the vacuum and pressurize the helium line while watching the PSI. Once the PSI reaches 1100, the rupture disc should break. Depressurize the chamber and remove the shot plate. The rice callus has now been bombarded and is ready for selection. Once finished, sterilize the equipment and prepare for your next transformation.